Good afternoon, everybody. My name is Dan Culver. I'm a lung doctor at Cleveland Clinic. I want to thank you for joining this afternoon. Today, we're going to talk about Efzofitimod, a novel agent that's in trial for sarcoidosis. Thanks for joining. This is a treatment that recently had a phase 1b/2a trial published and is now in a phase 3 trial. I want to point out that this is the first medication that's gone to phase 3 for any manifestation of sarcoidosis. And so hopefully this will actually be the first medication that receives registrational approval in this space in the era of randomized controlled trials. And so, of course, this is partially developing a new pathway for registrational design in sarcoidosis, but also hopefully it's really going to result in a therapy that helps our patients. I want to also note that I'm unpaid for any of this work that I do with aTyr. I believe in the field, but I'm not receiving any compensation for any of this talk or other things that I do with this company. So what is Efzofitimod? This is based on tRNA synthases. When I was in medical school, I learned that tRNA synthases were machines that help put protein together, and that is the canonical function of these proteins. But they actually also have non-canonical functions in which splice variants are secreted into extracellular space, and they have immunomodulatory properties there. This was an entire area of science that was discovered by a man called Paul Schimmel, who was the founder of this company, aTyr. And so when you disrupt either pharmacologically or by genetic means these extracellular tRNA synthases, you end up with some diseases. This area of biology hasn't been much explored, to tell you the truth, but it is quite conserved in an evolutionary sense. Many organisms use this to help modulate immune responses. And so what I'll show you today is a little bit of what we know about tRNA synthase biology and its target, which is Neuropilin 2 receptor. And I want to point out that this is not really completely well-defined science yet. We're learning as we go. So back to clinical for a moment. We had a trial that many of you have probably seen that looked at the safety of Efzofitimod in pulmonary sarcoidosis. And you can see the design here. This is a multiple ascending dose placebo-controlled trial in a small number of patients. There were some endpoints also focusing on efficacy, including steroid sparing, lung function, and patient-reported outcomes. But the main purpose of this trial was really to look at the safety and tolerability of the medication. I want to turn your attention to the light blue triangle. This is the steroid dose, and after 2 weeks from the start of the medication, steroids were tapered. This is a design that we initially thought wasn't very good for sarcoidosis, but more recently, I think this is really where the field is going, and I expect that future clinical trials in sarcoidosis are going to follow this template of steroid tapering. And there's two big reasons for that. One is that it helps unmask the difference between placebo and active medication. The second thing is that it may be an endpoint in and of itself. And I think that if you talk to patients, they'll all point out to you that steroids are relatively despised by patients, that they cause a lot of side effects that are both noticeable in the short term and that are sneaky and that cause long-term morbidity or mortality in the long term, like cardiovascular disease, diabetes, and obesity. So this is the new paradigm for clinical trials in sarcoidosis, steroid tapering. So 37 patients were randomized and dosed in the trial. You can see that there were a number of dropouts. And I want to point out that most of those were due to study sites closing or to patients not coming in during COVID. So I think the fact that this trial even got across the finish line is impressive in and of itself. The fact that it's a positive trial is even more impressive, and this is one of the reasons why I personally am fairly excited about the possibilities for this medication. And so all of the analysis I'm going to show you was based on intention to treat, and you can see that the intention to treat group really were people who got at least several doses of the study medication and then that completed a follow-up visit. And so this is now the set that we're going to look at for the next several slides. So the first bottom line is that there were no strong safety signals with Efzofitimod. It looked to be fairly well tolerated. We had a number of patients here in Cleveland that went in the trial. They didn't really notice any particular side effects. The only person that seemed like they got an infusion reaction actually was in the placebo arm getting saline. Here's the steroid reduction in the medication arm compared to placebo. You can see that we measured steroid dose in several different ways, and you can see that patients coming in on an average around 12 or 13 milligrams of prednisone were all able to drop. Even the placebo patients over the span of 24 weeks were able to drop their steroids on average, but that the drop was better and it was more sustained in the numbers on higher doses of Efzofitimod. One might say that this is not a huge difference, 7.17 versus 5.62, but I think it's important to remember that it takes a while for patients to start to redevelop their granulomas once they stop steroids, and also that you're talking about a relatively short time period. So as you think about the cumulative dose of steroids over time, even a difference of 5 milligrams per day in a prednisone dose might be pretty important if you're coming in the door on 7.5 or 10 milligrams. We really don't know how aggressively steroids can be dropped. In fact, with longer doses of medication that's effective, including potentially this medication. Here's the vital capacity data. You can see compared to placebo that there were in the 3 and 5 milligram per kilogram dose arms improvements in vital capacity. This is despite reduction of the prednisone dose. One might also say that this is relatively a small effect. And I would agree that it's not a huge effect and that it crosses zero. So it's not statistically significant, but the trend is a dose response. And in fact, the magnitude of the effect is not too much different than what was seen in the magnitude of the original infliximab randomized controlled trial. So again, I think that in terms of that physiologic endpoint, that this is all going in the right direction. Perhaps the most exciting endpoint is really the patient-reported outcomes. Here are 5 patient-reported outcomes: transitional dyspnea index, the Leicester Cough questionnaire, the fatigue assessment scale, and 2 domains of the King sarcoidosis score. The shaded area represents the numbers that exceed the minimal important clinical difference, and the asterisks denote the ones that are statistically significant, even with the small numbers. Again, the same theme emerges: a dose response, a consistency of effect. So whether or not this will hold up in a larger trial remains to be seen, but the early returns are certainly very promising. I also want to point out that steroids have been associated with negative quality of life before, and so we need to untangle whether some of the effects are due to simply the reduction of corticosteroids. Nonetheless, I think that this really is the paradigm where future clinical trials will go. And the reason for that is that we have to unmask differences in biology. If we only keep patients on baseline therapy of 10 or 15 milligrams of prednisone, along with some other immunosuppressive medications, it'll be very hard to see incremental benefits of any potential new medication. It's also important as you think about clinical trial design to recognize that not everybody's going to deteriorate during the course of a clinical trial. Probably up to around 50% of patients can stop steroids and over 6 months or even a year can just sit there looking perfectly good. What this means to us as clinicians is that we probably have patients who could theoretically be off of steroids today. It's just hard to predict exactly who that is. And that really to get the power to see the signal, we really have to do an active withdrawal of steroids across a study. I think the best really controlled data on this is the golimumab ustekinumab trial, and you can see the numbers there. The other thing that I always like to point out is that this really aligns with clinical practice. When we start any kind of new medication, we always withdraw steroids as part of what we're trying to accomplish when we're giving a new medication for patients whose disease is already controlled. One of the things that I think we fail to do in sarcoidosis is both endpoint and study design, and this steroid withdrawal mechanism really helps with this. Unfortunately, we've been a little late to the game to figure this out. And so because of that, there are very few data in sarcoidosis. So here are 5 diseases. Some of them are no more prevalent than sarcoidosis. In the yellow are the number of trials, and randomized controlled trials. and in the blue are the number of any clinical trials. And you notice a few things right away. Sarcoidosis is far behind the other diseases, and we have far fewer randomized controlled trials. And there are a number of reasons for that. One is that we've been a little bit intellectually sloppy with how we approach this. But more importantly, we really haven't developed good study designs, good clinical endpoints, and really good preclinical models of sarcoidosis. I want to point out that we do have some preclinical models of sarcoidosis, but we've been reluctant to say that these are decent models, even for granuloma formation. And so it's really hurt us in places like study section, or hurt us with sponsors thinking about, can I really study my medication or my target before I take it into patients? And we as a field or a community have really said, well, we don't like any of our models, so don't come and talk to us. So this is why you see these kinds of graphs with this great disparity in the number of trials that are happening. Now, if you think about how sarcoidosis happens, that'll help us inform future clinical trials. And so I'll talk a little bit about mechanism now. Here's how I think about sarcoidosis. There's some sort of an exposure. There's some sort of risk factors. Maybe a lot of it's genetic. And those two things come together. And there are also modifier exposures that might condition the chance of you reacting to the triggering antigen. And there's probably modifying genotypes that condition your chance of having that immune response. If we think about the immune response, this is really an antigen-driven, cell-mediated immune response. There's some sort of antigen here, and it's presented by a T cell, or sorry, by an antigen-presenting cell to a T cell. And we think that this is a very Th1 biased disease. And so you see a number of cytokines elaborated here, including interferon gamma, which are heavily associated with Th1 type responses. There's activation and proliferation of T cells. There's recruitment of more cells. Eventually, there's differentiation of monocytes into epithelial histiocytes and even fusion into multinucleate giant cells, and you end up with a granuloma. As you think about the complexity of what makes a granuloma, you realize there's a lot of targets for thinking about sarcoidosis biology and how we might treat it. But I want to point out this red oval in the middle. This is the antigen. And really, the problem is that for many, many years, we thought very carefully about what is this antigen? If I can just figure out what's triggering this forest fire, if I can figure out what caused the spark, then I can prevent it. And we didn't really pay any attention to why is the forest dry or how could things spread. We only worried about what was the spark. And I think now there's a growing conception that it doesn't really matter. There's probably more than one thing in the world that can trigger this. And really, maybe it's more the host and it's the immune response that we should be focusing on as we think about the biology of this. I also want to point out as a take-home message that we've really thought about this as entirely the adaptive immune response. What aspects of the immune response are dependent on that antigen presentation and on that oligoclonal T cell population? And as we look more at the biology, we see that there's a lot of interplay between the adaptive and innate responses. And so I think the innate immune response really is getting a resurgence. And we're really looking at some mechanisms that have more to do with the innate immune response and how we condition that, rather than worrying about an adaptive immune response, which might be important very early, but might not be as important as you get into more chronic disease. So this is a very simple cartoon. Let me show you a slightly more complex one. This is a network analysis in the top left from sarcoidosis lymph nodes, and it just looks at what gene networks are overexpressed in sarcoidosis. And you can see in the middle there is STAT1. That is the canonical signaling target of JAK signaling, which is what happens with interferon signaling. I told you already, interferon gamma is a key mediator of sarcoidosis and Th1 immune responses. And this is the dominant gene network in sarcoidosis. If you look at phospho-STAT1 on the bottom left, you can see it's highly expressed in sarcoidosis. This is one of the reasons why everybody's excited about JAK inhibitors now. This is going to go right at the heart of the pathway. And here on the right is just a network map of RNA-seq data from skin biopsies of patients treated with JAK inhibitors, autophagy for cutaneous sarcoidosis. And you can see that in that analysis, there are a number of JAK-STAT-dependent and JAK-STAT-independent mediators, which are proposed to be important in sarcoidosis. And I want to point out that several of these are interdicted by the molecule we're talking about today, and several of them are also the targets of other clinical trials that are happening. Well, Th1 is a nice hypothesis. We know the immune system's not quite that simple. And in fact, there's emerging data that suggests that more than Th1 cells are involved in sarcoidosis, and in fact, that there's plasticity of these cells. So I think when you read the sarcoidosis literature and you think, wow, I've got this nailed, I characterize the immune phenotype, just remember that as the immune system evolves from acute into chronic, into fibrotic, there's probably shifts in what's happening here with the immunology, and we need to be sensitive to which area of the disease we're studying, what era of the disease we're studying. And so there's an emerging evidence that there are deficiencies in Treg biology in sarcoidosis, and that there's an important role, or at least something of a role, for Th17 as well. Whether or not that's a therapeutic target, I think we won't get into today, but there's a novel thing that's been discovered in sarcoidosis, and that's really that it's not necessarily correctly characterized as a Th1 disease. The cells that produce most of the interferon gamma are actually midway between Th17 and Th1, so they've been labeled as intermediate T cells or Th17.1 cells. So Th17.1 is probably the main thing that we're looking at here in terms of the T cells that are helping drive this disease. The other thing this brings up to me is that, again, we talked about how the disease has different immunologic phenotypes as it goes into different phases. And so as we get further on to understanding the biology, we may think of this not as sarcoidosis, but as sarcoidoses, where there's a syndrome, an immunologic syndrome. These are two patients that I see. The one on the top left is doing great. He's actually in wonderful shape, does all kinds of exercise. He has sarcoidosis. The one on the bottom right is really symptomatic. He's in trouble. He has sarcoidosis as well. And in fact, the granuloma in the middle is from chronic beryllium disease, which is an autoimmune reaction to HLA modifications induced by beryllium, but which could be a version of sarcoidosis if one thinks about the number of antigens as a broad concept. And so I think as we parse these out more, we're going to get more precision into our treatment strategies. So just to sum up that part, I want to point out that the etiologic trigger here is probably not the main driver. That really what you're talking about is a black box of genes, epigenetic changes, host factors, and modifier exposures that set the stage. They make the forest dry, or whatever metaphor you want. Sarcoidosis develops. And then a black box of the same sorts of things, but maybe with different variables, genes, epigenetic changes, host factors, and modifier exposures, leads to different phenotypes. So this really de-emphasizes the etiology, and perhaps it de-emphasizes the importance of the initial adaptive response to focus more on the whole totality of the immune response. And so here's a cartoon that's very complicated, but it really demonstrates after an antigen presentation happens, activation of macrophages, formation of a granuloma, and then a number of outcomes that can include antigen clearance with remission, antigen persistence, either with fibrosis or without fibrosis, and then a whole bunch of potential targets that can play into this, and that all may be both mechanisms for future therapies, but many of these are actually interdicted by neuropilin biology. I also want to point out that the immune system, as I said, goes through some phases, and that one of the phases that's emerging as probably an important aspect of why sarcoidosis persists is this idea of autophagy. And so this is cells being able to resolve inflammation. And there are a number of diseases where autophagy has important roles with various triggers. And you can model this in several different models. There are also genetic studies looking at this. And this paper from Pacheco really is a nice exposition of the role of autophagy in sarcoidosis. And there is a relationship of autophagy to another molecule that's been shown to be important in sarcoidosis, which is this mTOR, mammalian target of rapamycin, in this TSC2 knockout mouse. If you look at BAL cells, and this is a really complicated plot on the right, but again, autophagy in human BAL cells has important roles. So how does that play into the molecule we're talking about today, Efzofitimod? Let's pivot to that for a minute or two. I won't show you any more of these plots, I promise. So we didn't know what Efzofitimod was doing before. And in fact, Efzofitimod is really a fragment of histidyl tRNA synthase, which binds to Neuropilin 2, which is a receptor I never heard of before this came up. As you start to look around in biology and look at what is NRP2 doing, it actually heterodimerizes with several other receptors like plexins and VEGF receptor, and it's a target then for semaphorin signaling, for VEGF signaling, and it has these domains as depicted here on the right side, and you can see that Efzofitimod actually binds to 3 of these. And just to jump to the punchline here, I'm going to show you data suggesting that Efzofitimod does not antagonize VEGF signaling or semaphorin signaling. So luckily, it doesn't look like it's going to have necessarily off-target effects here. Efzofitimod binds very selectively for Neuropilin 2 in a screen of about 4,500 cell surface receptors. There was no other binding partner identified for it. You can see that the EC50 is pretty low at 30 nanomolar. And here's a control that you'll see in a number of these next slides, N15. And you can see that that does not bind with any great affinity, even at very high protein concentrations. It also doesn't bind Neuropilin 1. So Efzofitimod binds the propeller structure that's formed between alpha 2, beta 1, and beta 2 domains of Neuropilin 2. With no real big binding to any single one. On the top, you can see the amount of binding in a luminescence assay really requires all those 3 domains. If you knock out some of those, like just having beta 2, beta 1, or beta 1 alone, you can see that the response rate is flat across different concentrations of Efzofitimod. And again, here you can see a different experiment that demonstrates the same thing. You really need all 3 of those domains to bind Efzofitimod. So in human biology, neuropilin is found in sarcoidosis granulomas. Here's immunohistochemistry in the middle. For those of you who don't look at these normally, the horseradish peroxidase stains brown. So when you see this brownish tint, that's where the neuropilin is binding on this immunohistochemistry. And if you just look at fish immune cytohybridization with immunofluorescence, you can see that the CD68, which is the macrophage and epithelioid histiocyte marker, is where you're finding the neuropilin 2. All those white things that look like a constellation are right there in the epithelioid histiocytes. So this is really in those innate immune cells where you're seeing most of this. In normal biology, as you differentiate from monocytes into macrophages or into dendritic cells, and NRP2 expression on the cell surface goes up quite a bit. So here are the experiments on the top, taking human monocytes, activating them, differentiating them into macrophages on the top or dendritic cells on the bottom, and looking at the shift in the amount of neuropilin 2 found on the cell surface receptor on this flow cytometry plot on the right. This looks like it tracks mostly with M1 differentiation of macrophages, which is, I know, a bit of a controversial topic because M1 and M2 are somewhat plastic and it's a little bit of an artificial distinction. That being said, many of the cytokines and drivers of sarcoidosis seem to have M1 features, and I'll go into that in just a second. So if you take sarcoidosis patient monocytes and you look at neuropilin 2, what is it doing? On the left-hand panel, are neuropilin receptor expression on monocytes from sarcoidosis patients. You can see it's higher. You can see that those same cells are secreting CCL2, TNF, and IL-6. We know that already in sarcoidosis biology, so that's a nice confirmation that we're getting the same readout. But if you look at our clinical trial, you see that Efzofitimod really reduced the levels of all of those, again, in a dose-response way. So CCL2, TNF, and IL-6 are remarkably reduced in the 5 mg per kg group. And so this is what I told you before, that Efzofitimod is not likely to interfere with other NRP2 ligands. Here's a luminescence assay. When you bind the heterodimer here on the left, it triggers a signal and there's a luminescence readout. And so if you look at ascending doses of efzofitimod, that's this blue triangle over here, and you just look at the amount of luminescence over time, you can see that even with ascending doses of efzofitimod, that you don't really blunt that curve of VEGF signaling or of semaphorin signaling by any real dose of efzofitimod, which is within the range that we would be using at all. So that's a nice, I think, reassuring finding. What does efzofitimod do to monocyte differentiation? It does look like it changes the macrophage phenotype. Here are two human donors. Just looking at the amount of macrophage spreading, you can see there's an effect from efzofitimod, and that also impacts NRP2 expression. If you take monocytes and differentiate them into macrophages and then look at gene expression in those using RNA-seq, you can see a number of pathways that are downregulated. And just these two green bars are each only one patient each, but you can see some pathways that are starting to emerge. So putting this all together, sorry, one more slide, and then we'll put it all together. Again, looking at expression in these differentiated macrophages, you see lower CCL2, lower TNF after treatment with efzofitimod, and you see changes in the cell surface expression, suggesting that the phenotype of these macrophages is changing. So CD14 expression goes down quite a bit when you differentiate cells under efzofitimod. That's the green over here. And this plot in the middle is showing basically the same thing as what you see right beside it, that CD14 is markedly downregulated on those cells, and that's really, I think, a marker of macrophage phenotype as those cells develop, and also of CCR2. So here's putting it all together. So here are a number of data looking at both cytokines and at receptors. Across the top are the various places where data has been checked, and you can see that there are clinical data. There's gene expression using RNA-seq in those differentiated cells that I showed you. There's cell surface receptor staining, and then there's protein secretion. And you can see that some of these were not done, some of these were not significant, but that many of the things are consistently found across a variety of these markers, again, that we think are probably important in the pathobiology of sarcoidosis. Efzofitimod probably has effects that are important in non-sarcoidosis diseases as well. I'll show you just one slide on this. And this is an acute lung injury model. This is a big hit on the immune system. And it's looking at the ability of efzofitimod to decrease cell recruitment into the lung. So this is an LPS installation model, intratracheal installation. And you can look at macrophages, dendritic cells, inflammatory monocytes or neutrophils. And you can again see a dose response on reduction of those cells trafficking into the lung. So I'm sure that this is going to lead to other sorts of studies looking at efzofitimod for other indications besides sarcoidosis. Stay tuned for that. So let me just pivot in the last couple slides to what's happening now, and then I think we'll have some time for questions. Here's the current trial. And this is, again, a registrational trial. It's a multinational trial. The idea is to take patients with moderate to severe sarcoidosis. You can see there's about 260 patients in this trial. And to really recapitulate the same study design as before, but with a slightly longer period of treatment. And so there's a steroid tapering phase over 12 weeks. The patients can come in actually with a little lower baseline dose of steroids than before, and importantly, not a huge fibrosis burden. And the primary endpoint here is going to be the reduction in steroids. And I think that this is really important because not only does this align with what patients are looking for, and it likely aligns with risks for long-term morbidity and mortality, but it's also a very more easy endpoint to measure with more likely responsiveness to an active, effective drug than something like vital capacity, which tends to be relatively flat and tends to be obscured by other things like obesity and fibrosis and airflow obstruction. So this steroid burden, I think, is going to be a very nice endpoint. And it's nice to see regulators getting behind this idea that this is an important thing. And I would say that this is really more in line with the way we think about asthma trials, and the likelihood of treatment failure or exacerbation or need for steroids as being important endpoints. And rather than thinking about sarcoidosis as analogous to IPF, where we're looking at some kind of inexorable decline in vital capacity as a useful endpoint. There are some key secondary endpoints. You can see a couple of them listed here, and there will be a bunch of exploratory endpoints, of course. The other thing I want to point out about this trial is that this is really the first good shot on goal in sarcoidosis during my career, and people are going to be watching. And look, at the end of the day, I want this trial to be successful. I want this medication to work. I hope it does, for our patients' sake, for the community's sake. It's going to make the world better if this drug works. But equally important to that is that we demonstrate that we can conduct clinical trials in sarcoidosis. There are other sponsors looking at this as well, and they're looking to see if there's a regulatory pathway that's going to work. And so as a community, we have to pull together and make this trial work. I think that this is a test case for other sponsors, frankly, and it's incumbent on us and our future patients and our future colleagues and our trainees to really try to make the field better by finding a pathway, so we can be like those other bars on that graph I showed you, where there are multiple players in the field that are interested in our space. So I'll get off my soapbox now and just conclude by saying that the take home here is that Neuropilin-2 is highly expressed in sarcoidosis, granulomas, and macrophages. It's a very conserved biologic process that we're just starting to understand now. But I think it's exciting and it's fun to learn about. Efzofitimod exhibits high selectivity for Neuropilin-2, and it has in vitro effects that really influence a number of key regulators of sarcoidosis immune responses. We have a small trial, but positive in terms of a dose response, positive in terms of consistency of endpoints, and apparently not associated with any significant side effects in pulmonary sarcoidosis. And so now we have this phase 3 trial I told you about. There is a little bit of a link here if you're interested in being involved in referring patients, or you want to learn more about the trial. Please email this number here. And again, I'm happy to address any questions about this. And it's been quite a pleasure to talk to you today. So I'm going to end the slideshow, and I'm happy to take any questions that you have. So I'm going to go from the chat. The first question is, do you think this drug has the main efficacy in early stage of pulmonary sarcoidosis, or can it be of value in a fibrotic stage, such as along with nintedanib? That's a really great question. I don't know the answer. I know that in the early stages, we didn't really necessarily see a strong correlation between duration of disease and the efficacy of the medicine, but the numbers are so small, I think it's just purely speculation to talk about this right now. If we think about what Efzofitimod and Neuropilin biology does, many of the things that have been studied so far have been shown to be present and upregulated in acute sarcoidosis, but also in chronic sarcoidosis. So I wouldn't necessarily say that chronic progressive sarcoidosis is going to behave better or worse to this medication. Of course, in acute sarcoidosis, many of those patients will undergo a spontaneous resolution. And so that's, I think, part of the reason why we require at least some months of persistent sarcoidosis in order to qualify for the trial. I also think that's part of the reason why we can recognize that even after 6 months, patients who require therapy and who have symptoms are quite unlikely to just spontaneously resolve over the next 3 months. So I think it's fair and reasonable to put those patients in. If you look at the biology, there are some data, preclinical data entirely, that suggest antifibrotic effects and potential antifibrotic effects of this mechanism. I think it's too early to completely tease that out, but it is certainly titillating to speculate that this might be a really great mechanism to treat those combined fibroinflammatory diseases like fibrotic sarcoidosis, like hypersensitivity pneumonitis, like scleroderma, like RA-ILD, all of those ones where you have those combinations of both of those. maybe you really get to hit two birds with one stone. I think there's a lot more work to be done before we can really be confident to make a statement like that. So please take that as a bit of speculation right now. Thank you for the question. So thanks for the nice deliberation. What are the baseline tests needed to be done before prescribing this biologic? Right now, of course, we can't prescribe it. It's only available through the trial. And just like any other trial, the main focus is on patient safety. And so there's a number of baseline safety tests that happen. Most of those are pretty familiar, routine sorts of tests like EKGs, basic blood work, pregnancy screening, pulmonary function tests. And then, of course, as you go along, many of those same studies are repeated. It's not my sense right now that there will necessarily need to be more sophisticated or unusual testing done in order to use this medication. I don't think that things like PET scans or advanced cardiac imaging are necessarily going to be part of the equation. But right now, of course, there will just be a proscribed set of tests based on what's in the study protocol. Thank you for the question. Here's a question from Ramona H. Does the drug inhibit the expression of Neuropilin 2 and then alters monocytes? It's a great question. I don't know that we understand yet exactly what it does to Neuropilin 2, whether that's on the transcriptional level or the translational level or some other level. The data that I showed you and that I've seen suggests that Neuropilin 2 expression does go down under treatment with the medication. So there may be some kind of feedback mechanism. But I don't know more. Don't know more than that. The next question: is a TB test needed? I don't think it will be. Certainly, you would screen for tuberculosis in treating these patients the same way you would screen for TB in any other patient undergoing immunosuppression. I don't think that this is likely to have the same kinds of effects as we saw with the TNF antagonists. But I think that we'll have to continue to monitor for infectious complications as we get into the larger trial. And hopefully we won't end up where we were with the TNF antagonists, where we had a surprising discovery of TB. I don't think this is going to have the same kind of granuloma busting thing where latent TB is released as what we saw with the TNF antagonists. Next question, can European patients be enrolled in the trial? Absolutely. There's a number of sites in Europe. I'd refer you to the prior email address, and maybe I'd ask if somebody can put that email address back in the chat. That would help. But you can find all of the sites if you just email to that address. Absolutely, a number of the patients will be enrolled in Europe. Got a question. Where would the assumed role of Efzofitimod fall in the algorithm of sarcoidosis management in the presence of other DMARDs, such as methotrexate or infliximab? It's a great question. You know, I think everybody would like to know the answer to that. I'll give you my perspective, but you have to remember that I'm just one person, and that other people are going to get a vote here. For example, payers are going to make some decisions about what has to happen before you might get this medication. That includes government payers and insurance companies, for example. This will also be something that the patients will have a vote on. And finally, people who are dealing with this medication are going to get a handle on it and understand what the role is. So I think some of the considerations that are going to influence are going to be the efficacy, the tolerability, the durability of the effect, the ease of administration, the cost, and I think that whether or not this does something to sarcoidosis and other organs is also likely to be an influencer of the uptake, assuming that it even gets approval, right? Like we're assuming that this even makes it across the finish line. But if it does, all those things are going to play into it. I think one thing as a field that we can do right now is that we can continue to hammer home the message to regulators and sponsors, and especially to insurance, that long-term steroid treatment is not good for our patients. Long-term steroid treatment is sneaky, and it seems like it's okay until your patient comes in the door and they've got diabetes, or they need a cataract surgery, or you've altered their metabolism, so now they have a heart attack. And I think we have been very slow in the pulmonary space to recognize that as an issue. And so the idea that, well, it's just 7.5 of prednisone and it's just fine, I'm not going to worry about it. We got to get away from that thinking. And we got to find agents that really allow us to act just like the rheumatologists and get to a much lower dose of steroids or off steroids altogether. So where it falls in the algorithm is going to depend on a lot of factors, but I don't think it's okay to just say, well, everybody needs to be on steroids and this is only for people who fail steroids. That may or may not be the right approach. Another question, will it be available in an oral form or is it an IV infusion? Right now, it's an IV infusion. Sanjay or somebody else may want to weigh in on whether there is any development of any other route of administration, sub-Q or something like that. That might be something coming in the future. But right now, what's happening is that this is being studied as an IV infusion, and that would be the initial approval for that. Sanjay, do you want to comment any more about other routes of delivery?
Sure. I'd say right now IV is clearly how we're proceeding with the drug formulation. However, life cycle improvements could include subcutaneous administration in the future.
Thanks, Sanjay. Okay, what are the criteria for failing oral corticosteroid tapering in the study? What are the criteria? So in order to taper the steroids, there has to be on both the patient side and the physician side a perception of improvement or at least of stability. If you meet both of those things, then there's a continued taper. If either of those things are worse, then you don't taper, and there's an option to actually increase the dose of steroids. I don't recall that there are any distinct criteria in terms of like change in vital capacity, change in a PRO that absolutely mandate how you behave. And the idea, I think, is really to try to mimic as closely as possible the way we think as clinicians. And that is that we take all the information available to us, PFTs, radiography, what the patient tells us, what the exam sounds like, how well the patient walks into the exam room, the 6-minute walk test, if you have that. We put all that together when we make our decisions. And so I think coming up with single dichotomous criteria that say, you know, in this situation, you must go up, and in that situation, you must go down, it's a little bit pseudo. And so I think this idea of putting that all together into a global assessment is really what this is trying to emulate. Of course, all that stuff's being captured. Another question, can you tell us more about the patient population targeted in this study? Again, these are patients who have symptomatic treatment requiring, coming in on treatment, pulmonary sarcoidosis, you know, without another major organ that's driving the treatment indication. So patients who have treatment requiring cardiac sarcoidosis or neurosarcoidosis or ocular sarcoidosis would not be good candidates for it. These are patients who have symptoms and who have at least 6 months of disease. So again, we're de-emphasizing pulmonary function testing here. We're, of course, trying to exclude patients with very much fibrosis. And so there is a requirement about not having too much fibrosis. But in general, it's a pretty wide net, and it's designed to really give us, you know, more generalizable data that we can then take into patient populations. The exclusion criteria are predictable. Besides the things I mentioned, there are exclusions for severely impaired PFTs, substance abuse, issues with being able to follow the protocol, concomitant diseases that are significant, all the things that you would normally think about. Schedule of administration is once a month, IV. Should also mention, while we're waiting for more questions to come into that, as we think about these ways of treating sarcoidosis. We've adopted many things from rheumatology and from some other specialties, but mostly rheumatology. And we've done so on the basis of case reports, and there's nothing wrong with that. We've really brought a number of good medications into the field, which have helped our patients and helped us reduce steroids. That being said, I think that we're now moving past that. And I think we're moving to a stage where we need to have more nuanced and strongly evidenced biologic rationale for either importing drugs, repurposing drugs into sarcoidosis, or even for our clinical trials. And so I would ask all of us as people in the field to really continue to focus on the science, focus on understanding how different agents impact granuloma biology, how we can get into a precision medicine approach to thinking about what we're doing with these agents. And I think there was a question already about what phase of sarcoidosis this would fall into. This is going to be a very important piece of what gets measured here. So I think that that's my plea for supporting science and supporting a scientifically driven approach to therapy. And thank you, Sanjay, for pointing out and reminding me that there are a number of sites in Japan that are also participating in the study. And so 60 centers, 260 or so patients. Plenty of opportunity for people to find a place to enroll in the trial. There are centers all over the place. Okay, well, it seems like we've run out of questions here. I'm going to go ahead and thank everybody for the opportunity to speak today. And thank you for your attendance and your interest today. And I'm going to wish you all a very nice day and a great rest of the week. Take care.